Like the lecture last week, this week's lecture will meet the course objectives by first identifying prevalent communicable diseases and their current epidemiology and routes of transmission in Canada. Secondly, recognizing their control and prevention by current public health policies, practices, or regulations. Thirdly, recognizing relevant laboratory techniques and tests used to culture, identify, and classify human pathogens and relevant terms encountered in reports or in the literature. And finally, understanding the major mechanisms used by microbial pathogens to cause disease. This week, we'll look at communicable diseases that are transmitted by direct contact and respiratory routes in Canada, which are influenza, the common cold, SARS and COVID, invasive pneumococcal disease, tuberculosis, and invasive group A streptococcal disease. We'll also be able to recognize the clinical features of these diseases and comprehend their pathogenesis, as well as identify the transmission routes and epidemiology of these diseases, recognize laboratory methods used to confirm their diagnosis, and recognize how the transmission of these diseases may be controlled and prevented. Looking at the incidence rates of diseases transmitted by direct contact and respiratory routes over the past decade, pertussis tops the list, as we mentioned last week. We also saw that the incidence rate of invasive pneumococcal disease last week was between 9 and 10.9 cases per 100,000 population. The other two diseases that are also marked by a red asterisk are the ones we'll be covering today. Tuberculosis and invasive group A streptococcal disease both have lower incidence rates at around 5 cases per 100,000 population, about half the rate of invasive pneumococcal disease. We'll also be looking at flu, SARS, COVID-19, and the common cold, all of which have incidence rates that are not reported as annual rates in the conventional sense, due to their occurrence as either past or present outbreaks, epidemics, or pandemics. How many of you have had the flu? You probably didn't know for sure that it was flu and never got tested. That's why most respiratory illnesses that have flu-like symptoms are called flu-like illnesses or ILIs. Clinical symptoms usually have an abrupt onset after a short 2-day incubation period with fever, chills, headache, and muscle aches, which peak in severity around 6 to 12 hours later. The infection then progresses from the upper respiratory tract to the lower parts involving the trachea, bronchus, and lungs. A dry, non-productive cough usually develops, and symptoms can persist for 3 to 5 days. There's usually significant improvement after more than a week. However, fatigue, weakness, and coughing may persist for as long as 2 to 6 weeks, and in a small number of cases, the illness can become life-threatening due to secondary bacterial infections. Complications can arise from bacterial superinfections with Streptococcus pneumoniae, Haemophilus influenza, Staphylococcus aureus, because the infection with the influenza virus can be so severe that it has created a situation where the immune response is now exhausted. This is especially the case in most susceptible individuals, such as the elderly. Many of these bacteria are present as normal flora and don't normally cause disease. But after a bad bout of flu, it is possible to then end up with a secondary bacterial infection that is tough to fight even in young, healthy individuals. It is not surprising that life-threatening cases of flu are more commonly reported in the elderly, individuals with underlying medical conditions, such as chronic cardiac or pulmonary disease. Many severe cases of flu develop superinfections with bacterial pneumonia that becomes life-threatening rather than an overwhelming viral pneumonia, that is a rapid progression from the original influenza virus infection. Like most respiratory diseases, flu is spread directly from person to person through respiratory droplets or aerosols, which are much smaller and able to travel further than 6 feet. Flu transmission is seasonal and usually peaks during winter months. High-risk groups include school-aged children, residents in congregate living settings, such as long-term care facilities, seniors' homes, and correctional facilities. Workers in high-exposure risk groups include healthcare workers, for example doctors, nurses, and dentists, who perform aerosol-generating procedures such as bronchoscopies and invasive specimen collections. Laboratory workers who handle cultures from known cases are also at high risk. Let's look at the case definition for influenza before delving into more epidemiology, as this enables us to know what the criteria are for identifying a confirmed case of influenza. Based on the Canadian National Surveillance definition, a case of influenza can be confirmed by 4 different methods. This can be by isolating influenza virus from an appropriate clinical specimen through culture, or demonstration of influenza virus antigen in an appropriate clinical specimen, or determining that the IgG antibody titers in the convalescent sera of the case is at least 4 times higher than those present in the acute sera. Aside from serological tests, nucleic acid tests can also be used to detect influenza RNA. Canada's National Surveillance System monitors the spread of the flu and other flu-like illnesses on an ongoing basis. Since not all cases of influenza are tested in the laboratory, influenza-like illness is monitored across the country by a network of laboratories, hospitals, doctors' offices, and provincial and territorial ministries of health using clinical symptoms such as the acute onset of respiratory illness with fever and cough, and with one or more of the following: sore throat, joint pain, muscle aches, or fatigue, which is likely due to influenza. Gastrointestinal symptoms may also be present in children under 5, and fever may be absent in the elderly and children under 5. Part of the Canadian Influenza National Surveillance System, or FluWatch, involves monitoring outbreaks in schools and workplaces. If there is greater than 10% absenteeism rates due to cases with influenza-like illness, specimens should be collected and sent for testing, especially at the beginning of the flu season, as it may be the first indication of community transmission in an area. Flu outbreaks in hospitals, residential institutions, and other settings are defined as 2 or more cases of influenza-like illness within a week, with at least 1 lab-confirmed case, and these should be reported within 24 hours of identification. Residential institutions include, but are not limited to, just long-term care facilities and prisons, as they may also be retirement homes and other congregate settings. As I mentioned on a previous slide, FluWatch is the Canadian National Influenza Surveillance System, which monitors current and retrospective influenza activity levels across the country. Unlike the calendar year, the flu season starts on week 35 and ends on week 34. For the 2019-2020 season, this corresponds to August 25, 2019 to August 22, 2020, and the dates change every year. Unlike the National Surveillance System, the flu season dates used by Public Health Ontario and Toronto Public Health are different, and the dates do not vary from year to year. They always start on September 1st and end on August 31st. So there's a little bit of difference between provincial and national surveillance data. I've added the epidemiological curves from 2018 to 2021 on the next few slides to show the seasonal incidence of flu and also what it looks like in a typical year. The 2018-2019 influenza season in Canada was longer than the previous 5 seasons and was characterized by 2 waves of influenza A activity and very little influenza B circulation. The national season started in week 43, that is October 21st to 27, 2018, peaked in week 52, which is December the 23rd to 29, 2018, and ended in week 21, corresponding to May the 19th to the 25th, 2019. H1N1 influenza A was predominant in the earlier part of this season, from October to February, followed by a smaller wave of H3N2 influenza A circulation from March to April. H1N1 influenza A cases are the dark red bars and H3N2 the lighter red bars. Over half the influenza A specimens were not subtyped. There was also a much lower incidence of influenza B at the end of the season from April to May, and this is shown by the blue line. The 2019-2020 flu season was very different and ended 9 weeks earlier than the past 5 seasons. An abrupt end was also observed in several countries. This coincided with the implementation of public health measures to reduce the spread of COVID-19. Testing for influenza and other respiratory viruses has been influenced by the current COVID-19 pandemic. Changes in laboratory testing practices from March to June may also affect the comparability of data to previous weeks or previous seasons. In the 2020-2021 flu season, in spite of the elevated levels of testing, there were only 69 positive influenza tests, which is significantly lower than in the previous 6 seasons during which more than 50,000 cases were detected. One major reason for this is the same for the truncated flu season observed in the previous year. That is, public health interventions to prevent the transmission of COVID-19 have been very effective in preventing the spread of influenza. It is interesting to note that 31 of the cases were actually associated with a new live attenuated influenza vaccine, and they do not represent community circulation of seasonal influenza viruses. Even though live influenza vaccine strains are attenuated, they can still be recovered by nasal swabs following vaccination. I've not been able to get the national incidence rates for influenza, and all I have are the number of cases per year, which is around 50,000, if we look at the numbers over a 2-year period from 2018 to 2020. Ontario Public Health data indicated that over the past decade, the incidence rate of influenza was between 14 to 133 cases per 100,000 population. This corresponds to just under 2,000 and 20,000 cases a year. The latest data available was for the 2019-2020 flu season, in which there were less than 10,000 cases. In Toronto, the incidence rate of influenza in the past decade ranged from 29 to 157 cases per 100,000, so it is slightly higher than the provincial average. This corresponds to between 790 and 5,000 cases a year. This is the epidemiological curve of influenza over the past decade for Ontario and Toronto. As I mentioned on the past slide, the incidence rate in Toronto is slightly higher than the Ontario rate. The incidence rate also increased over time in both Toronto and Ontario. If we look at the epidemic curves in Ontario in the same flu seasons as Canada, we can see that they're very similar. The 2018-2019 flu season shows a typical bell-shaped curve, with flu A being the predominant serotype and low numbers of flu B till the later end of the season. For the next flu season in 2019-2020, the same truncated curve observed for all of Canada was also present in Ontario. Basically, the percentage of positive results for both flu A and B dropped to baseline levels much earlier than in previous flu seasons. The bars represent tests carried out indicating that the low numbers of positives were not due to lack of testing, but was a real result due to the public health interventions implemented for COVID-19. This is the latest data we have for this province up till the end of last year. Although it looks as if the number of positive tests have increased, if you look at the y-axis, you'll notice that on the very right, the scale for the percentage positive tests actually just goes up to 1%, whereas on the graphs in the previous year, it was between 20 and 22%. So this graph is not comparable to the ones in past years. And if the y-axis on the right was plotted as 20%, instead, the lines would be at baseline level. The laboratory diagnosis of influenza involves the collection of nasopharyngeal or throat swabs during acute illness. Viruses are then cultured in mammalian kidney cell lines, such as the Vero cell lines from an African green monkey, or MDCK, Nadine Darby canine kidney cell line from a Cocker Spaniel. Hemagglutination assays are carried out to determine the viral titers. Immunological assays, such as immunofluorescence and serology, can be used to either detect viral antigen in the respiratory tract secretions from epithelial cells, or a greater than 4-fold increase in antibody titers between convalescent and acute serum samples collected 10 to 14 days apart using hemagglutination inhibition assays. There are three major serotypes of influenza viruses, A, B, and C, based on their ribonucleoprotein antigens. All influenza viruses have single-stranded antisense segmented RNA. Influenza A and B have 8 segments, whereas C has 7 segments. All influenza viruses are enveloped and have glycolipid membranes which are derived from host cells. There are two different spike proteins for all serotypes of the influenza virus. The first one is hemagglutinin, which enables the virus to attach to host cells, and the second protein is neuraminidase, which enables envelope fusion as well as viral release from host cells. In the diagram, the hemagglutinin spikes are blue with sharp points, and the neuraminidase spikes are green with knobs. Influenza A has many different hemagglutinin and neuraminidase subtypes, and the combination of different H and N subtypes can potentially result in 198 different ones. However, only 131 different subtypes have been identified so far, and there are only a small number of subtypes that infect humans predominantly. These are the ones that have the H1 to H3 and N1 to N2 subtypes. The most common flu A subtypes isolated from humans are H1N1, H3N2, and H2N2. Aside from humans, influenza A can also infect other animals, and it can be a zoonotic pathogen, especially when pigs get infected. Infections can be severe and seasonal outbreaks occur quite regularly. The reason influenza outbreaks occur is the ongoing mutations in the hemagglutinin and neuraminidase genes during viral replication. These mutations are subtle and sometimes result in only one amino acid residue change. However, this may cause antibodies from previous infections and immunizations to lose their neutralizing effect and become less protective. This subtle change in antigenic type over time is known as antigenic drift. Due to the presence of many different variant strains, a naming system is in place for them. The first character is the serotype, so it can be A, B, or C, then followed by the geographical origin, the strain number, the year of collection, and the hemagglutinin and neuraminidase subtypes in parentheses. All these parameters are separated by forward slashes. Due to its many different subtypes and also the ability to infect a range of animals, influenza A viruses can recombine genetically if an animal host becomes co-infected with different subtypes simultaneously. I have provided an example of a pig that is infected with H1N1, then gets co-infected with H3N2 from a human, likely the farmer. This could result in 4 different subtypes, including 2 new ones, H1N2 and H3N1. When this occurs, the new viruses may have major changes in their hemagglutinin and neuraminidase spike proteins. This is known as antigenic shift. This effect may happen all of a sudden and is very difficult to monitor or predict. Generally, it's not a matter of if, but when. Due to the rare nature of recombination events, antigenic shifts are much less frequent than antigenic drifts, usually every 8 to 10 years. Large populations around the world will not have any prior exposure or immunity to these new flu viruses, and pandemics can occur. This includes the 1918 Spanish flu, which is the one most people have heard about, but there are also more recent ones, such as the 2 pandemics that resulted from Asian strains, and the most recent pandemic in 2009 that many of us have survived. I've also included the estimated global case numbers and mortalities to show the impact of these pandemics. Through genetic characterization, the 2009 H1N1 influenza virus was found to have a complex lineage. It was determined to be a quadruple reassortant virus, meaning that it contained virus genes that originated from 4 different sources. Some of the gene segments originated from North American swine flu viruses, some from North American bird flu viruses. 1 gene segment originated from a human flu virus, and 2 gene segments were normally found in swine flu viruses from Asia and Europe. Laboratory testing also showed that the 2009 H1N1 influenza virus did not have any of the 1918 pandemic flu virus genetic markers associated with increased risk of severe disease, nor markers that were previously associated with high death rates in people infected with the avian influenza A H5N1 virus in other parts of the world. The first known human cases of avian flu were reported in Hong Kong in 1997. The virus had the H5N1 subtype. There were 18 cases, 6 of which were fatal. The virus has mutations in 2 RNA segments in the genes for the PB2 polymerase and hemagglutinin, which resulted in increased virulence in humans. Human-to-human transmission of avian flu is low. Unfortunately, outbreaks have not occurred, although most cases involve close family contacts. Bird flu is acquired through direct contact with infected birds and cannot be transmitted by eating infected birds. Over the past 20 years, there have been more human cases with different subtypes, although H5N1 remains the predominant subtype. Others include H7N2, H7N3, and H7N9. Cases have been reported in Canada, with the first H5N1 case in North America reported in 2014. The case, who resided in Alberta, acquired the infection in China. H5N1 flu is a highly pathogenic avian influenza and has a high case fatality ratio, and this case did not survive. The outbreak in 2004 involved a large number of poultry farms in British Columbia and the H7N3 subtype of avian flu, which is a low pathogenic avian influenza. The 2 human cases developed eye infections and mild respiratory symptoms. However, 19 million birds had to be euthanized. Influenza B is not as complicated as influenza A, as it only infects humans. It is most stable antigenically, and antigenic drifts are observed, resulting in seasonal variations. Since it doesn't undergo antigenic shifts, it doesn't cause pandemics. It may cause severe infections on occasions and outbreaks every 4 to 5 years. Influenza B viruses have 2 main lineages, the Victoria lineage and the Yamagata lineage, both of which are included in seasonal flu vaccines. Influenza C can infect both humans and pigs, but the infections are usually mild. Outbreaks are limited and viruses vary due to antigenic drift. The influenza virus uses its hemagglutinin spikes to bind sialic acid receptor sites on human respiratory cell surfaces. It then invades and multiplies in ciliated respiratory epithelial cells, causing localized inflammation and cytotoxic effects on neutrophils and alveolar macrophages. The damaged respiratory epithelium is highly susceptible to invasive bacterial superinfections unless recovery is mediated by interferon production and cytotoxic T-cell lysis of virus-infected cells. Neutralizing antibodies are also protective. These are antibodies to the hemagglutinin spikes, which can neutralize flu viruses when a second infection occurs. Antibodies to the neuraminidase spikes also limit the spread of viruses in the host. Although antivirals to treat influenza are available, they need to be used early in the illness to be effective. These include ion channel blockers, such as amantadine for flu A, and neuraminidase inhibitors, such as oseltamivir for both flu A and B. For most milder cases of flu, supportive therapy is usually sufficient, with rest, fluids, analgesics, and over-the-counter cough-suppressing medication. NACI, or the National Advisory Committee on Immunization, recommends the seasonal influenza vaccine for all Canadians aged 6 months and older, with a focus on individuals at high risk of influenza-related complications and their household contacts. These include several groups listed on this slide, but the one I would like to point out is the elderly, as there is a different vaccine that is recommended. This vaccine contains a high dose of hemagglutinins from 2 flu A strains and 2 flu B strains. The seasonal flu vaccine is also recommended for individuals capable of transmitting influenza to those at high risk, and these include healthcare workers and other care providers, as well as those who provide service in closed settings and essential community services. Individuals involved in avian influenza-infected poultry culling operations should also be vaccinated. Influenza immunization programs using seasonal flu vaccines occur every year, just before the start of the season. Vaccines usually contain killed or split virions, but some are also live vaccines. The main vaccine component is the hemagglutinin spike protein. Annual revaccination is required due to the different strains that circulate, and the vaccine aims to be a good match with these strains in order to provide good immune protection. There are usually two types of flu vaccines, a trivalent formulation that has two flu A and one flu B strains, and a quadrivalent formulation with two flu A and two flu B strains. In some seasons, poor vaccine efficacy is seen when the match is poor and only partial protection is obtained. When we look at the 2018-2019 flu season, the latest season with an annual report, we can see that there was a good match between the circulating and vaccine strains for both the two influenza A subtypes. Aside from hemagglutinin inhibition assays to determine the type of strains that were circulating during the 2018 to 2019 flu season, genetic characterization was also carried out for the H3N2 strains by sequencing. This genetic analysis determined that most common subclades observed was the 3C2A1, which matched the vaccine strain. This match between circulating and vaccine strains was also observed for the two influenza B subtypes, although there were more of the Victoria lineage strains circulating than the Yamagata lineage one. While the vaccine efficacy, which is the level of protection that the seasonal flu vaccine provided to the immunized population, was greater than 50% for both the H1N1 influenza A subtype and influenza B, and 56% overall, with a higher coverage than previous years. What was really odd was that the vaccine efficacy was unusually low for the H3N2 subtype. This anomaly has been observed in previous years as well. It has been reported that this is due to antigenic drift in egg-based cultured vaccine strains, which makes the mutated strains variant to the original strains, thus not conferring the anticipated protective immune responses. More genetic variation was also observed in circulating H3N2 strains than in other flu subtypes. For the first time this year, the seasonal flu vaccine will no longer contain viruses that have been cultured in eggs. Instead, cell-cultured viruses will be used. How many of you have had a cold? If I could see you raise your hands, I bet it would be most of the class, if not all. This is probably why the term common is used as an adjective. There are two viruses that cause the common cold. Rhinoviruses cause more than half the colds, and coronaviruses to a lesser extent. The common cold is a respiratory tract infection. Clinical symptoms include sneezing, runny nose, and eyes, rarely fever in young children. It is not a life-threatening illness, but may lead to sinus and middle ear infections. Colds are spread directly through respiratory droplets, or indirectly via soiled hands and articles freshly soiled by nasal secretions from infected individuals. Transmission tends to be seasonal, usually during non-summer months. It is more prevalent in preschool children and gradually declines with increasing age. Humans are the only reservoir of infection. Rhinoviruses belong to the Picornaviridae family, which is actually pico for small RNA. They are naked, single-stranded positive-sense RNA viruses, and there are 115 classified serotypes. The host cell receptor is a glycoprotein called ICAM1, which stands for Intercellular Adhesion Molecule 1. Rhinoviruses can be cultured in human fibroblast cells. In spite of many years of research, there are currently no vaccines or drugs available for the common cold. Generally, drinking hot, soupy fluids is recommended to ease symptoms. The other group of viruses that can cause colds are the coronaviruses. These are enveloped single-stranded positive-stance RNA viruses. Coronaviruses have spikes on the envelopes that give them a solar-like appearance. Today, we're familiar with the ones that cause pandemics such as SARS and COVID. However, many coronaviruses don't cause severe disease, even though there are two main outbreak-causing strains that circulate and have caused disease outbreaks in long-term care facilities. These are known as 229E and OC43. Some of these outbreak strains are able to cause life-threatening illness in the elderly, and an outbreak of OC43 was identified in British Columbia in 2003, just after the SARS pandemic. SARS, in comparison, was a novel coronavirus in 2003, and it emerged for the first time, and there was very little information available on this new coronavirus. In fact, it took a while for SARS to be identified as a coronavirus. You can see that it has different host cell receptors to the other coronaviruses that cause common colds. Which brings us to SARS, which is caused by a coronavirus. I hope you remembered that we looked at how SARS arrived in Toronto through an infected traveler. The symptoms are flu-like but can progress to more severe life-threatening pneumonia with shortness of breath or acute respiratory distress syndrome. SARS is transmitted from person to person by close contact with an infected individual, either by caring for or living with them, or being in direct contact with respiratory secretions or bodily fluids. Viruses are transmitted readily through respiratory droplets when infected individuals cough or sneeze. For mice contaminated with infectious droplets such as elevator buttons and carpets have been reported to play a role in disease transmission, especially in the hotel in Hong Kong where the index case was staying. The SARS outbreaks in Toronto resulted in 44 deaths and 351 cases, the majority of whom were healthcare workers or those associated with healthcare institutions, such as patients and visitors. A fifth were household members of cases, and a small minority were associated with community events and travel. Because of this predominant association within the healthcare setting, SARS is a communicable disease of public health and occupational health concern. SARS was the first coronavirus pandemic that most of us became aware of. Some of you were very likely young at the time in 2003, which was more than 17 years ago, so were blissfully unaware of the health crisis that hit the GTA at the time. However, it was bad enough that many RAS and public health students did not get a final letter grade in 2003 because many of the public health inspectors teaching the courses were too busy dealing with the outbreaks. All they got on their transcript was a pass or fail. There were 29 countries all over the world that had at least one case of SARS, and in total, there were just over 8,000 cases, many of whom were healthcare workers, and a small percentage associated with travel. The global case fatality ratio at the end of the outbreak was estimated to be approximately 11%, based on the high mortality rates in the elderly. The basic reproduction number, R0, which is the average number of secondary infectious cases produced by an infectious case, was estimated to be around 3 in the absence of specific public health measures such as isolation. This figure was based on the calculations of 3 different research groups. So where did SARS come from? Surveillance for bat SARS coronaviruses in the wilderness areas around Hong Kong and China indicated that the type of bat called the horseshoe bat was carrying coronaviruses that were very similar to the SARS coronavirus. Genome sequencing of these bat isolates and phylogenetic analysis indicated that the bat SARS coronavirus sequences clustered with human SARS coronavirus sequence. Based on other sequencing studies and also epidemiological investigations on the environmental origins of the SARS virus in China, it was determined that the source of the infection came from wild animals that were sold for human consumption at a market in southern China. Viruses isolated from 2 animal species, the palm civet and raccoon dog, were found to have a close genetic relationship to the novel human SARS coronavirus. Their antibodies were also able to inhibit the growth of SARS coronaviruses isolated from humans and vice versa. One of the main studies that determined the intermediate animal hosts of the SARS coronavirus sampled a range of wild animals that were sold in the market in southern China that was associated with the SARS outbreak index cases. They isolated the virus from palm civet cats and raccoon dogs, as well as detected neutralizing antibodies. There was a 99.8% homology between the full genome sequences of animal and human virus isolates. However, phylogenetic analysis of the spike gene grouped the animal and human sequences into 2 different clades. This indicated that some of the 0.2% difference was located in this gene. There was also another main difference, a 29 base deletion that was present in human virus isolates. This large deletion supports the evolutionary pathway of the novel SARS coronavirus from animals to humans, as the likelihood of losing 29 nucleotides far exceeds that of gaining such a large stretch of extra genetic material. This significant mutation of the SARS coronavirus in humans has been shown to promote virus replication and program cell death of host cells. The paper also reported higher exposure to the SARS coronavirus in different occupational groups using antibody neutralization and immunofluorescence assays. They found that 40%, that is 8 of 20 wild animal traders, and 20%, that is 3 of 15 individuals that slaughtered animals in the market, were seropositive, compared to only 5% of vegetable traders, that is 1 of 20, and none of the controls, who were hospital patients with non-respiratory diseases. This study was supported by another study on cases in a SARS outbreak that occurred in 2004, after the main pandemic was declared over by the WHO. Once again, exposure to palm civets was found, and 2 of the 4 cases included a waitress and a customer of a restaurant with palm civets on the menu and on the premises. 5%, that is 2 of the 39 restaurant workers, had antibodies to the SARS coronavirus. They were the head waitress who often helped customers to select palm civets from their cages and a cook. As I'm unable to share the first phylogenetic tree due to copyright reasons, I thought I should show you the neighbor joining tree from the second study. The spike gene sequences from the palm civets in the restaurant and the index case all clustered together on the same branch, and I've surrounded them with a red box. In the blue box below, on a separate branch, are 2 spike gene sequences from palm civets found in the wet market in southern China associated with the original SARS outbreak in 2003. You can see that molecular epidemiological studies using genetic variation in virus mutants are very useful in tracking disease transmission. Either specific polymorphic genes or whole genomes are targeted and sequenced. The products are then analyzed by DNA sequencing or genetic fingerprinting using restriction fragment length polymorphisms or other molecular methods. When the first cases of SARS emerged, there were no laboratory tests available because it was such a new virus, so many of the cases were reported as suspect or probable. Today, with several tests available, and nasopharyngeal aspirates or swabs and stool specimens can be submitted for testing. Nucleic acid tests using reverse transcription PCR or serology can be used. The criterion for a seropositive result is a 4-fold or greater increase in antibody titers between acute and convalescent serum collected more than 8 days apart, using a number of serological tests such as ELISA or immunofluorescence assays, Western blotting, or virus neutralization assays. The results need to be confirmed using at least 2 different clinical samples or 2 different laboratory tests. All newly identified sporadic cases have to be verified by a WHO SARS reference laboratory. SARS can be prevented by using personal protective equipment. This is especially important in a healthcare setting, where N95 or other certified masks, goggles, and face shields need to be worn together with gloves and gowns, as well as using good hand hygiene practices. Suspect cases need to be isolated in single occupancy rooms or separated by more than 1 meter apart if this is not possible. Negative pressure ventilation and exhaust systems should be used with more than 12 air exchanges per hour. There should be an independent air supply. All equipment and surfaces should be disinfected. Contact tracing is critical. Contacts should be actively monitored for 10 days by public health and advised to self-monitor for symptoms such as fever. During epidemics, there should be good communication with the public, media, as well as other health professionals. There should be adequate triage facilities and their locations publicized so that suspect cases can be diverted there for screening and care. All cases are required to be reported to the World Health Organization. The SARS pandemic came and went, and it wasn't until 2012 that another novel coronavirus outbreak was reported in the Middle East. There appears to be an association to bats, with camels as the intermediate host, as they have high antibody levels to the virus, but the definitive reservoir host has yet to be identified, as viral genomic sequences are not similar enough to provide any association. So, we've arrived at the last viral respiratory disease, COVID-19. I'm sure most of you are familiar with the symptoms, and the symptoms listed here are actually identical to the ones for SARS. What I've done that I think is useful is to draw up a table comparing the symptoms of 3 of the respiratory diseases in this lecture. When we look at the list of symptoms, we can see how close flu and COVID resemble each other, so it makes it very difficult to diagnose these diseases clinically. Colds, on the other hand, are quite different, so it's actually possible to tell if you just have a cold rather than COVID. However, since the symptoms of COVID can vary from person to person, what may seem like a mild cold initially may sometimes turn out to be COVID. Also, as we know well now, there are quite a number of cases that are asymptomatic, and that's perhaps the most dangerous group because these individuals have no idea that they are infected, go out and about to run errands or work, then end up infecting other people in the process. This is where routine screening can be useful in preventing workplace transmission. COVID-19 is caused by a coronavirus that is closely related to the 2003 SARS coronavirus. It is actually more closely related to the SARS-like coronaviruses from bats than the 2003 SARS coronavirus. Once again, we don't have much information on the initial case in the outbreak in Wuhan, China. Some of them were associated with a large wholesale seafood market. However, the intermediate host animal has yet to be identified or confirmed, although genomic sequence similarities have been found in SARS coronaviruses from Malayan pangolins. The picture on the right shows you what they look like. They are scaly anteaters. We looked at the number of cases of COVID-19 in the current pandemic over a month ago. However, the massive numbers continue to climb, and more than 100 million cases have been reported since then. The regions here are WHO ones. Currently, the United States still remains the country with the highest number of cases in the world, with more than 75 million cases and close to 1 million deaths. In Canada, we've had more than 3 million COVID-19 cases, with more than 35,000 deaths. This is an increase of around 500,000 cases over a month since we last looked at these numbers in Lecture 2. Unfortunately, since then, 5,000 more people have lost their lives to this disease. Currently, Ontario has the highest number of cases, followed by Quebec. That's because most of the Canadian population is concentrated in these two provinces. Since the population varies so much in Canada, it is more accurate to compare incidence rates than raw case numbers. You can see the geographic distribution better on the graph on the next slide. This is a map showing the provincial and territorial rates, which is a better indicator of actual disease incidence, since the population distribution across Canada is so varied. You'll notice that even though Ontario has the highest number of cases, it actually has a lower incidence rate than many other provinces, such as Alberta and Quebec. As of February 21, 2022, more than 1 million confirmed cases have been reported in Ontario. The highest number of cases were in adults between 20 and 39 years of age, and the lowest number of cases were in preschool children below the age of 5. The next lowest number of cases were reported in seniors over the age of 80. This group unfortunately suffered a disproportionate number of deaths compared to young children as well as other younger Ontario residents. High mortalities associated with COVID were also reported in residents of long-term care homes, most of whom were seniors. Outbreaks were reported in more than 1,000 retirement homes and nearly 2,000 long-term care homes. While hospital outbreaks were usually limited to a small number of cases, long-term care homes generally had large numbers of cases and deaths. Some facilities lost as many as a third of their residents. Even though more than 8,000 cases were reported in healthcare workers in long-term care homes, only 10 deaths have resulted. Being elderly is therefore one of the highest risk factors associated with increased severity and poor outcomes for COVID cases. In Toronto, while information for the majority of cases was not obtained, nearly a quarter of the cases somehow acquired their infections while out and about in their community and did not have any contact with a known case. Other cases were acquired through close contact with another case or household contacts. Less than 5% of the cases were travel-related. The rest of the cases were exposed in either healthcare, congregate, or institutional settings, which includes long-term care, nursing or retirement homes, shelters, and correctional facilities. I thought it would be interesting to look at where the workplace and community outbreaks were occurring in Toronto, You can see from this table that the setting with the highest number of outbreaks is in the manufacturing, distribution, warehousing workplace. Massive outbreaks with more than 200 cases in Canada Post sorting and Amazon distributing facilities have resulted in a number of shutdowns and demonstrate how workplace disease transmission can be rapid and widespread. Other major workplace outbreaks were associated with office settings and construction. In terms of community settings, most outbreaks have been associated with restaurants and bars. Nasopharyngeal or nasal swabs, as well as saliva specimens, can be submitted for testing. The main test for COVID-19 is a nucleic acid test using reverse transcription PCR to amplify 3 different genes, which are the spike and nucleocapsid protein genes, and the open reading frame 1AB. Rapid antigen test kits are also available for at-home testing. In terms of treatment, Health Canada has authorized 2 different therapies for COVID-19. The first is a monoclonal antibody to the spike protein called bamlanivimab, which blocks attachment and entry to host cells. And the second is an antiviral, which is a nucleotide analog called remdesivir, which is used to treat severe cases of this disease. Public health interventions to prevent transmission are what we hear through the media on a regular basis. I've listed them, but won't repeat them since I think everyone is very aware of them. At present, we're on the verge of abandoning nearly all of these measures and are relying mainly on vaccination to protect us. Let's look at the approved vaccines. The first 2 are both mRNA-based ones, the Pfizer and Moderna vaccines. The AstraZeneca vaccine is more conventional, as the vector has been used in other viral vaccines, such as the ones for Ebola and Zika, and unlike the mRNA vaccines, doesn't require any special storage conditions, just refrigeration. However, it's no longer being administered in Canada. Nearly 80% of the Canadian population, meaning more than 30 million people, are now fully vaccinated with 2 doses of vaccine, and more than half of the individuals who are 18 years or older have received a booster shot. One of the variant or mutant viruses that has made the news is the UK variant, which is in the lineage B.1.1.7. It was found as part of a genomic study in September 2020 and shocked the investigators with the unusually large number of mutations present. 8 of the mutations are in the spike protein gene, in the part that codes for the receptor binding domain. This has resulted in increased binding to the host angiotensin-converting enzyme 2 receptors, immune evasion in immunocompromised individuals, and enhanced respiratory cell entry. One of the mutations in the spike protein gene causes a change in the spike protein cleavage site, shown to promote entry into respiratory epithelial cells. This variant has higher transmissibility and has been associated with increased risk in disease severity and death based on 3 separate studies carried out by leading academic institutions in the UK, including the London School of Hygiene and Tropical Medicine, Imperial College, and the University of Exeter. The second variant of concern that has been reported is the South Africa variant, which has even more mutations in the spike protein gene. This has caused increased binding to the host ACE2 receptor, as well as reduced antibody recognition. This variant has been associated with a higher viral load and higher transmissibility. As far as we know, there have not been any changes in clinical presentation or disease severity in cases infected with this variant. Although since this particular variant is newer than the UK one, studies have yet to be carried out to determine if this is really the case. In terms of vaccine efficacy, a study using plaque reduction neutralization tests has shown that there was no reduction in neutralization antibody titers of sera from Pfizer-BioNTech vaccinated individuals, so it appears that the mRNA vaccine or any vaccine that uses the spike protein should work for now. The third variant of concern is the P1 variant from Brazil. It has been designated by the WHO as the Gamma variant. It was detected around Christmas 2020 when a large outbreak occurred in Manaus, the largest city in the Amazon region. The same mutations are present in the spike gene as the UK and South Africa variants that cause increased binding to the host ACE2 receptor as well as reduced antibody recognition. The reason it is called the P1 variant is a matter of technicality. Because viral lineages cannot have more than 3 numbers or sublevels in total, a new alphabet was selected, and P was chosen based on pangolin. However, this variant really belongs to the B1128.1 lineage. What is particularly worrisome about this variant is the scale of the outbreak, which is the second one to occur in this area, after the first large outbreak which occurred in the earlier part of 2020. This indicates that the immunity gained from a previous infection is not protective against this type of new variant. The Delta variant was first reported in India in late 2020. Like the other variants of concern, it also has a number of spike gene mutations that make it more transmissible, in fact at 50% greater than the Alpha or UK variant. It also has a higher R0 than other variants of concern and has been reported to cause more severe disease. Since immune protection from vaccines and previous infections is reduced, it is more likely to cause breakthrough infections as well. Omicron is the most recent variant of concern. It has a number of sublineages. BA1 is the prevalent variant in the current pandemic wave that resulted in a sharp increase in cases at the end of last year. There are such a large number of mutations in the spike gene that this target is not detected in PCR tests. It is highly transmissible, and even immunizations with boosters is not sufficient to prevent infection. Fortunately, most breakthrough infections are mild, and vaccines offer protection from severe illness. There is also another Omicron variant, BA2, which has started circulating. It is even more transmissible than BA1, and some early reports indicate that it may cause more severe illness than BA1. I thought we should look at the phylogenetic tree of these variants of concern to understand how diverse the genomics of the SARS coronavirus 2 is. In terms of diversity, both the Delta and Omicron variants have branches that are quite separate and distinct from the first three variants of concern, that is Alpha, Beta, and Gamma. The Omicron sublineages of BA1 and BA2 are also very diverse and do not share much homology with each other, which means that we will likely not have much immune protection after infection with either variant.